Help in Pharmaceuticals in association with Higher Secondary Principals Forum.
Hello everybody, welcome back to another lecture in Biomolecules. One part of the topic was discussed in the previous lecture, that is Biomolecules part 1, where we discussed carbohydrates. Now moving on. Okay, here today we are going to discuss proteins in this topic and nucleic acids. So let's begin. I mentioned also earlier in the previous lecture proteins. What are the sources of proteins? Like you can see, and very tempting also, the chicken, fish, any meat, then milk products like cheese, then the eggs, albumin in the eggs, it's a rich source of protein. Then where else do you find protein? Other than the sources that we take in protein, you also find protein in the different parts of your body, right, from head to toe. Anything and everything you name will have some kind of protein. Hair, the nails, hemoglobin in the blood, it's nothing but proteinaceous. Then the brains, the nerves, enzymes, all enzymes are proteins, right. The cellular constructions, they are the proteins. Antibodies are proteins, messengers, muscles are proteins, and anything and everything you analyze, you will find some kind of protein. Okay, so and that's very important. What is the role of protein? Basically, they are the building blocks. It helps your body to grow. It helps you to construct. Very, very important. So we are going to learn a little in detail about this proteins. So like I said, what are the sources? Milk, cheese and pulses, peanuts, fish, meat, etc. And it is also present, I said, in every part of the body, helps you to grow and helps you to maintain your body. The last line is very, very important. The monomer, that is the smallest units of proteins, are nothing but amino acids. So you have to know what amino acids are, and then some amino acids, because they are the primary constituents of proteins. What are these amino acids? Basically from the word, you know, amino acid, it's a very confusing word, you see, because we know there are two functional groups. One is the amino group and acid is a carboxylic group, and correctly if you have inferred that amino acids are made up of both the functional groups, amino functional groups as well as carboxylic acid functional group, which you see there are two different groups having two different properties. Amino, you know, is a basic group, whereas carboxylic, you know, is an acidic group. So both basic and acidic groups is present in amino acids, and thus making it amphoteric. We'll see how. Further, amino acids are classified as alpha, beta, gamma, delta and so on. Now I'll just mention to you what this means. Look at the picture. I'm not sure if it is too clear. What are alpha amino acids? Alpha means, now that's the structure, okay. Primarily what will it be made up of? A carboxylic group, some carbon atoms and an amino group. Now suppose the amino group is attached to the carbon that is adjacent to the carboxylic. So the first carbon, that is, you see this carbon, the first carbon that is attached to the carboxylic group, if that carbon carries the amino group, it's called as the alpha amino acid. Okay. Further, this side you see, the first carbon attached to the carboxylic group is called as the alpha, the second is called as the beta and so on. So if the amino group is present on the beta carbon, it will be called as a beta amino acid. And so this is the carboxylic group. The first carbon attached to the carboxylic group will be called as alpha, the next carbon is called as beta and the third carbon is called as gamma. So if the amino group is present on the carbon gamma, it will be called as the gamma amino acid. So basically what I am trying to tell you is how these are classified as alpha, beta, gamma. It is with respect to the carboxylic group. So if the carboxylic group, the carbon attached to the carboxylic group, the first carbon, if that is carrying amino, it is alpha. If the second carbon attached to the carboxylic group is carrying amino, it is beta, and so on. Proteins are all primarily made up of alpha amino acids, that is this kind of amino acids. Amino acids are of all these kinds, but proteins are made up of alpha amino acids. Some examples, there are many examples, I have just picked up 2-3, glycine, alanine and leucine. Amino acids are classified into two groups. One is essential amino acids and non-essential amino acids. Now essential amino acids, now you don't go by the word, okay, essential manje it's not, doesn't mean that it is needed, and non-essential doesn't mean not needed. So you don't have to go like that. Both are needed by the body, essential as well as non-essential. The difference is essential cannot be synthesized by your body. Okay, your body makes amino acids, but some which cannot be made by the body, they are called as essential. So obviously they will have to be supplied by the external source. So you will have to take those in your diet. Those are essential. And non-essential are the ones which you may not take by the through the diet, because they are the ones that your body will synthesize. So that's how you have essential and non-essential amino acids. Now there is something very interesting about amino acids. Okay, what is it? Now you see the amino acids we said is made up of both the groups, NH2 which is a basic group and acidic group, that is the carboxylic group which is acidic group. Now what's a base and what's an acid? We know that an acid is the one which will give away proton, whereas a base is capable of accepting a proton. So the molecule here is capable of undergoing self reaction within itself by losing and gaining the proton with itself. How? The COOH group is capable of donating a proton which will be taken by the NH2 group, and that is what is shown here. So this H will be lost and it will be formed COO minus, and NH2 which has a lone pair of electrons on nitrogen, will pick up that proton which COOH is losing and will form NH3 plus. So you see this molecule has both the charges, the negative as well as positive. Such a molecule which undergoes self reaction, self protonation, and it accepts proton within itself and forms an ion of this kind, having both the ends, the negative as well as the positive end, is called as a Zwitter ion. In amino acids, they are present as Zwitter ions. Very important word to remember what a Zwitter ion is. So in aqueous solution, amino acids will exist as Zwitter ion, which shows an amphoteric behavior, both acidic as well as basic, because it has both the groups. Now what is the structure? What is exactly the structure of proteins? We said that the proteins are made up of amino acids. Amino acid is the simplest repeating unit in the protein structure. They are called as the monomers of the protein structure, and precisely the alpha amino acids in proteins. Now 2 amino acids in proteins is bonded by a bond which is called as a peptide linkage or a peptide bond. Very, very important. How it is formed? By a condensation reaction. I have shown an example there of glycine and alanine. You see this glycine, both are alpha amino acids because the NH2 group is attached to the carbon very next to COOH. What happens is it undergoes condensation reaction with loss of water molecule. So what you are left with is a amide linkage, that is CONH. So you remove OH from here and remove one hydrogen from here, water, no, H2O, that means one hydrogen, one OH. So remove one OH from here, remove one hydrogen from here, and there is a new bond between CO and NH, which is called as the peptide bond or the peptide linkage. Now since there are 2 amino acids linked by a single peptide bond, such a bond so formed is called as a dipeptide bond. Di indicates 2. Dipeptide, you have to remember, does not mean 2 peptides. Dipeptide means 2 amino acids are linked by a single bond. So, similarly, if there are 3 amino acids, they will be linked by 2 bonds. So, that will be called as a tripeptide. Okay. Now, I am clarifying this because often the confusion is because of the name dipeptide. If you go logically, dipeptide you might end up thinking means 2 peptide bonds, but no. Okay, what is a dipeptide? When you have 2 amino acids linked by a peptide bond, one peptide bond, that is called as a dipeptide. So, you have one peptide but 2 amino acids. So, that is dipeptide. So, you have similarly tri, tetra, penta and so on. So, having more than 10, it becomes a polypeptide. So, if there are more than 10 amino acids linked by peptide bonds, it will be thus called as a polypeptide. Okay. So, a protein is a polypeptide having a lot of amino acids linked by peptide, and then doesn't end there. All these amino acids, all these polypeptides chain will undergo systematic further folding and thus giving the proteins a structure in the 3 dimension. Okay, we have to learn that further, that the backbone of this peptide chains will undergo foldings and they will thus give the proteins some kind of a structure. What structure? We are learning further. So, depending on that, depending on the structure, you have 2 types of proteins. One is fibrous. As the name says, fibrous will be fiber-like. Fiber-like is when the polypeptide chain runs straight parallel to each other. Okay, so, you have several spiral shaped polypeptide chain and they are bonded by maybe sulfide bonds, hydrogen bonds and so on. So, basically a rope like structure that run parallel to each other, and those are called as fibrous, the shape of it. Similarly, you have globular proteins. Globular proteins, as the name says, globule is spherical, right. So, when this polypeptide chain intertwined and coil around itself and form like a globule, they are called as the globular proteins. Okay, for example, here collagen, if you have heard of collagen, it is the largest protein in the human body. It is a fibrous protein, okay, because you have fiber like polypeptide chain running only parallel to each other and there is no coiling, whereas hemoglobin, myoglobin, they are globular proteins wherein they coil and form a spiral structure. Okay, so, this is depending on shape, depending on the structure, you have 2 types of proteins, fibrous and globular, simple. Further, there are stages that you can learn the structure of protein in 2, okay, like the primary, the secondary, tertiary and quaternary. You can learn them at different levels. Primary is the basic structure of the proteins. What is it basically made up of? You see, they are primarily made up of amino acid in a fixed sequence. There is a definite order in which this amino acids, maybe glycine, then alanine, leucine, all these are linked one after the other in a definite sequence. That becomes the primary structure. I am not talking about types of proteins now, I am talking about how you can learn the structure of protein at different levels. Primary means what is it basically? Basically, it is just a sequence of amino acids linked by peptide chains. Okay, so, this is what the primary structure means. Then comes the secondary structure. What are secondary structures? Secondary structures are those backbone of the polypeptide chain will interact with each other through bonds like hydrogen bonds or sulfide bonds and will undergo some kind of foldings. Okay. So, because of its secondary structure, there are 2 types of folding. One is called as the alpha helix, the other is called as the beta pleated sheets. from the words and the structure you will clearly understand what it is here. So, there is one polypeptide chain you can see, and what is happening is that polypeptide chain is simply coiling around itself. Now, why does this happen? You see there are the dotted lines that are shown here. The dotted lines that are shown, what is it? The oxygen from one spiral curve there forms a hydrogen bond with the hydrogen of the other NH from the other end, other spiral, and that is how you get a coil sort of. So, what will happen is, imagine you cut this bonds, okay, you cut this hydrogen bonds, what will happen? The spiral structure will uncoil itself. So, basically if you take any rope and you want to hold it as a spiral structure, there have to be some some kind of a force that will hold the turns, right? One turn, the next turn, the next turn. Now, here the bonds that are holding the turns are the hydrogen bonds. Why? From where are the hydrogen bonds coming? That structure only, the hydrogen of the NH, that is the amino group, and oxygen of the CO groups. Okay, so every time a CO and a NH forms a hydrogen bond, it will coil, and that is how you get this structure which is called as the alpha helix. So, a spiral structure, that is the secondary structure. There is another secondary structure which is called as the beta pleated sheets. Now, here you see the polypeptide chain, just think of they are running straight and they are coming back, imagine. There are some kind of bonds that will be holding this and this together, okay. By some kind of bonds I mean hydrogen bonds, the sulfide bonds, etc. So, you can clearly see it is the same polypeptide chain, there are no two polypeptide chains, just that it is going about parallel to each other and there are bonds that are holding this, therefore, thus forming like sheets, okay. They are not coiling, they are forming like parallel sheets parallel to each other, and that is the secondary structure. So, what is the primary structure? The arrangement of the amino acids in a definite sequence. And what is the secondary structure? How this polypeptide chain bond to each other through hydrogen bonds, and what is the resulting structure. Okay, it is either alpha helix or beta pleated sheets. The tertiary structure is the three-dimensional arrangement, okay, of the polypeptide chain. Further, how will it fold about and how will it overall fold and result, and that is how you get the shape like the fibrous and the globular shape, okay. So, further foldings rather. So, for example, now this typically arises because of the interaction of side chains, not just hydrogen bonds but not just sulfide bonds, there are so many other kinds of bonds. For example, there is this polypeptide chain that you can see, and these are the possibilities. What are the possibilities? One is here. There can be an ionic bond between a side chain that is coming from this side. This is a side chain. This entire group I can call as R of this part, and this is a side chain from this part. So, if they come close, meaning if the folding is such a way, there can be an ionic bond between the two. Then there can be something called as hydrophobic interactions, and there can also be hydrophilic interactions. Hydrophobic is water hating, you know. So, the groups that are water hating may be going away from away from water and coming closer, and likewise hydrophilic, some groups that are there around can be going towards water. So, because of all this interaction, that polypeptide chain will further take up shape, okay, will further fold, will further come close and all, and that is what results is then the tertiary structure. Okay, so there can be hydrogen bonds also, there can be disulfide bonds also, there can be hydrophilic or hydrophobic interactions, there can be ionic bonds also, and so on. So, with all this it will lead to the tertiary structure. So, what have we seen as of now? Primary structure is only the sequence of the amino acids. Secondary structure is how hydrogen bonds interact between the polypeptide chain and gives you only two types of structures, alpha helix and beta helix. Tertiary structure is how further this alpha and beta helix or the polypeptide chain through different interactions come closer and take up shape, which is a tertiary level. And then finally you have quaternary. Quaternary is when you compare the interaction between two or more polypeptide chain. For example, I have shown here 4 polypeptide chain, different colors, yellow, green, blue and light blue is taken so as to show the interaction between 4 different polypeptide chain with each other, and that is how you get the quaternary structure. So, basically a spatial arrangement of the polypeptide chain with respect to each other. 2 polypeptide chain with each other, 3 polypeptide chain with each other and so on. Yes, so what we just spoke about is this, that is the protein structure levels. One is primary, secondary, tertiary and quaternary. Primary is when it has sequence of amino acids. Secondary is because of hydrogen bonds, we have spiral or beta pleated. Tertiary is entire polypeptide chain thus folding over, and quaternary is 2 or more polypeptide chain, what happens with each other, how they interact and form bonds with each other and gives the final shape. So, that is about structure of protein. So, we have discussed 2 things as of now, sources of proteins that you are so familiar with, and next is structure of protein, what it is primarily made up of. Amino acids. Amino acids are bound together by peptide chain, and we can study the structure at 4 different levels. And also we have learned that there are 2 types depending on the shape, the fibrous and the globular. Now further, you must have seen these kind of phenomena. Okay? For example, the egg, the white albumin of the egg, you will notice when you subject it to different conditions, like you can change the temperature, you can put pressure, you can change the pH, what will happen is there is something called as denaturation of the proteins. Very, very important. What is this denaturation of the proteins? We said that the proteins can be spoken of having structure at 4 different levels. Denaturation means the structure of the protein gets destroyed. What structure? The quaternary, the tertiary, the secondary structure gets destroyed. What does that mean? How are these structures arising? How are the secondary, tertiary and quaternary structures arising? Because of foldings. How and why do they fold? Because of bonds. So, like I said, if you break the bonds, the structure will get destroyed. For example, in the helix, if you break the hydrogen bond, it will uncoil, right? The sheets that are running parallel to each other, if you break the bonds in between them, they will become anti-parallel, they will not be running like parallel sheets. So, with these conditions, by changing the temperature, by increasing the temperature, by changing the pH, you can actually break the bonds, and if you break the bonds, the structure gets destroyed, which is called as denaturation of proteins. However, you have to very well remember that it is only the secondary, tertiary and quaternary structure that gets destroyed because of bonds and not the primary. The primary structure remains intact. Okay, why? Because the primary structure is nothing but the amino acid sequence that cannot change with temperature and pressure. So, yes, when a protein is subjected to physical change such as pH, hydrogen bonds get disturbed, they rather get broken, because of which the globules will unfold, the helices will get uncoiled, basically the structure will be destroyed, and which is why the protein loses its biological activity, and this is called as denaturation of proteins. And during denaturation, like I mentioned to you, only the secondary and tertiary and quaternary structure will get destroyed, but primary structures remain the same. And one very common example is coagulation of the egg white, that is the albumin. You see the albumin getting coagulated when you heat it. And another example is curding of the milk. So just a fun picture that I have put there. I threw your protein cake away because the protein gets denatured. Something for you to ponder on why he is saying that. The other fellow is saying, you fool, do some research and stop replying on hearsay. So that's about proteins. Very little part of proteins is actually touched upon. The next thing is nucleic acids. Nucleic acids, which stores the very important genetic information responsible for heredity, passing on the the genes, etc., etc. So very important. There are two types of nucleic acids. If you have heard of DNA, that's deoxyribonucleic acid, which are double stranded, the one that I am pointing out, and RNA, which is called as the ribonucleic acid, which is single stranded. Okay, a little bit of nucleic acid we have to know. Now what is put at the side? Other bases. We are coming to that further. So DNA, this is just a picture I found very interesting, that it's a double stranded helix that run anti-parallel to each other from 5' to 3' and 3' to 5'. I am sure the bio students are going to be learning more detail about this. Just the chemical composition, what it is actually composed of, that is what we have to focus on. Okay, nothing more from here. Now, like carbohydrates, like proteins, like any other polymers are made up of smaller units, which are the monomers. Likewise, nucleic acids are also made up of repeating units, which are the monomers, called as the nucleotides. So the nucleic acids can also be called as polynucleotide. Okay, for example, we spoke about proteins. What is the repeating unit in proteins? The amino acids, that is the monomer. In proteins, the monomer is amino acids. Likewise, in nucleic acids, the monomer is called as a nucleotide. Therefore, because there are many nucleotides which are bonded, we will see by what bond further, let me not reveal that. So because all these nucleotides are bonded and form long chain of the repeating unit of nucleotides, they are called as polynucleotides. So what does a nucleotide consist of? What is it made up of? Very important. It has three major parts. Okay, you have to remember it like this. One is, it will always contain a base. What base? A nitrogen containing base, which is heterocyclic. The ones that were mentioned earlier here. These bases. That's one of the major component of a nucleotide. Okay, so there will always be a base. There will always be a sugar, which is going to be a 5-carbon sugar. If you have seen the previous lecture, the other part 1 on biomolecules, I have mentioned there also ribose. It's a sugar that is present in DNA. So the pentose sugar, because it's a 5-membered 5-carbon sugar, and there is a phosphate group. So all these three parts together make a nucleotide. Let's talk a little in detail about these. So what are these nitrogenous bases? So there are two types of nitrogenous bases, that is the purines and the pyrimidines. Purines are these, that is adenine and guanine. If you notice, purines, that is adenine and guanine, they are made up of two rings, fused rings. Okay, so they are clubbed together and called as purines, that is one is adenine, one is guanine, and pyrimidines are single rings, okay, which are uracil, thymine and cytosine. So, nucleotide may contain any one of these, okay, either an adenine, guanine, uracil, thymine and cytosine. So, basically they are all bases, with a nitrogen. So, it is a nitrogenous heterocyclic base. That is about the base. Next thing it will contain is a sugar. Now, the sugar we said is either ribose or deoxyribose depending on what you are talking about. If it is a DNA, it will contain deoxyribose, RNA contains ribose. Now, the structure of both you see is the same. The only difference is that is highlighted here, deoxy is having one oxygen less, whereas ribose has an OH. Rest all is the same. At carbon 1 you have OH, H, hydrogen, and here this is the only difference. On carbon number 3 you see there is a OH, H, carbon number 5 there is a CH2OH and hydrogen. So, that is the sugar that is present in a nucleotide. Okay, the difference is deoxy and ribose. Deoxy is because there is no oxygen and then ribose has an OH. Alright. So, three things again let me remind you that a nucleotide should be containing a base which is either a purine or pyrimidine or a pyrimidine, and also it will contain a sugar and it will contain a phosphate group. Now, in addition to nucleotide, there is something called as nucleoside. Now, what is a nucleoside? A nucleoside will contain only two things. One is the base, one of those bases, and it will contain a sugar. It will not contain a phosphate group. So, that is the difference between nucleotide and nucleoside. Now, how are these nucleotides containing these three things linked to each other? So, that becomes one nucleotide. So, that nucleotide is linked to another nucleotide by one special kind of a bond which is very, very important, and that is called as a phosphodiester bond. Very important, okay. A phosphodiester bond connects, what do you say, connects two nucleotides from between carbon number 3 of one sugar of 1 unit of nucleotide. You see, this is 1 unit of nucleotide having the phosphate group, having the sugar and the nitrogenous base. Now, I have just put a ring here and not shown you what that nitrogenous base is. That is not so important. What is important for me to show you here is the bond which is called as the phosphodiester bond here. Okay, that is the phosphodiester bond that links the carbon number 3, oxygen of carbon number 3 and carbon number 5, carbon number 3 of one sugar and carbon number 5 of the other sugar, called as the phosphod. There are two bonds basically, diester bond. So, there are two bonds that links the two units through carbon 3 and carbon 5. Another picture just to show the phosphodiester bond, how it links, and that is why you say that the chain runs from 5' to 3'. How is this? Okay, a phosphate group, a sugar, a nitrogenous base, but the sugar of one unit is linked to the sugar of the other unit. So, one unit of nucleotide is shown in green here, the other unit of the nucleotide is shown in red here, the third unit of the nucleotide is shown in blue here. They are all linked between carbon 3 of one sugar, carbon 5 of the other sugar through the phosphate group, that is through a phosphodiester bond. So, again here 3 and 5 through a phosphodiester. There are two bonds, you see here, oxygen, oxygen. So, there are two ester bonds actually. So, for diester, hence the name phosphodiester. This is what is very important for us, okay, in the chemistry point of view. The bond that is present that connects two nucleotides between what carbon and what is the bond called is the most important. A little bit of the structure of DNA now. So, the structure of DNA, it has, I mentioned earlier, two strands running anti-parallel to each other from 5' to 3' and 3' to 5', and they are coiled around each other through those bondings. And what do you say, the bases, you have to know what pairs with what. I am sure you must have learned this earlier also. Adenine will pair with thymine, guanine with cytosine, and so on. Okay, so thymine with adenine, cytosine with guanine, and so on. So that's the pairing of the DNA. RNA are single stranded, and there are three types of RNAs. One is mRNA, rRNA, and tRNA. Nothing in detail, I am just touching on the types of RNA. Nothing much to know about RNA. What you have to know is particularly the composition of DNA, that the DNA are repeating units of nucleotides. Okay, the monomer is a nucleotide, and the nucleotides are bonded to each other by a phosphodiester bond, running the diester bond between carbon number 3 of one nucleotide, that is of the sugar, and carbon number 5 of the other nucleotide of the other sugar. What is the biological importance of this nucleic acids? First of all, they are the reservoirs of the genetic information, and that is how genes are inherited, and different structural and appearance and everything passes on from one generation to another. Replication, you know that they are capable of replicating, and the RNAs are involved in protein synthesis. So your body can make proteins with the help of RNA. So that is all about nucleic acid. Because of this pandemic, a lot of and you don't have classes, regular classes, a lot of portion is deleted. I am sure you know about it. Just refer to your textbook and see which part of it is deleted, and you will know that there is more of it, like vitamins is deleted, enzymes is deleted, and disaccharides and polysaccharides are also deleted. So what you are left with only in this chapter is to study carbohydrates, proteins, and nucleic acids. Most important, look at it in the chemistry point of view, that is its composition and its bonding, and of course importance also you have to be knowing. So that is all. Thank you very much. I am hoping you have understood this topic. Thank you once again.
Prudent Scholars, powered by Lupin Pharmaceuticals.